We have Robyn Underwood with us, and I will unmute her in a moment. But she's going to talk to us. I don't know. It's the queen rearing stuff, correct? And so I think now I will just unmute Robyn. But before I do that, I just want to encourage people, if anybody is interested in helping us sell honey, I just sent out an email about volunteering. I put up a new volunteer sign up page. So if anybody would like to look at that and help with some honey selling in the near future, that would be great. Now I will stop talking, and Robyn, you may proceed.
Okay. I'm not actually talking about queen rearing, so I hope I prepared the right talk. I am talking about a queen comparison. And I got you preliminary, although today's latest data to show you. So let me tell you, you don't have sharing allowed.
Okay, you're the co-host now, so in theory you should be able to share whatever you want.
Okay. All right, you can see my slides now? All good? Okay. So the work I'm going to tell you about today is a collaborative effort of Penn State and Purdue University, along with cooperating beekeepers in each of our states, that's Pennsylvania and Indiana, and Liz Cambron, who did a bunch of the lab work, and a new person that I should have included here, Charles Dean, who is doing the current lab work. And he is going to be presenting this work even more up to date than what I'm going to be showing you at the state meeting during a poster session. So each day on Saturday and Sunday at lunch, there's going to be scientific posters, which is like a static version of a talk. Like at lunch, you can just go talk to the author, ask questions, look at the graphs and stuff, but it's a different format than giving a lecture. So you can talk to him there. He knows a lot more about the lab aspects of this project. So you know that there's like over 3 million colonies in the US, 125,000 beekeepers. We contribute to the US economy in a big way. And so the bees that we keep and the work that they do is valuable not only to us as beekeepers, but to, you know, the food that we eat. And when beekeepers are thinking about purchasing bees, there's a lot of different bees to choose from. In fact, I see on Facebook a lot new beekeepers always like, I'm looking for Italian bees. I must have Italian. And I was like, well, who told you that Italian is the best? And how do we even know? And then there are other beekeepers who've been doing it a long time and they want to improve their stock and are willing to pay a premium for specific stocks or breeds of bees that have claims behind them of performance. And the problem is sometimes they've never been evaluated in a side-by-side manner. So that's what we set out to do here, is look at how various stocks perform in our latitude. So Pennsylvania and Indiana have very similar weather and climate. And so we work together with beekeepers in our states to do this side-by-side comparison. So when you're thinking about what bees to buy or what stocks to recommend to other people, if I was in person, I would ask you to raise your hand and tell me which things that you look for or which claims would make you pay more for a queen. And so, of course, you know, most of us are in it for the honey. So you want to have some bees that are going to make you excess honey so that you can share it with friends and family or make enough to sell. And definitely so that it's not, you know, October the 20th and you are wondering if you fed them enough, right? So you want them to make enough food for themselves and excess for you. You also want to have a good time. You don't want to be miserable and all stung up every time you go out to your bees. You don't want your friends and helpers to be afraid to come and help you. And you don't want to be a danger to your neighbors. So you don't want to have bees that are too aggressive. So a lot of times the top two traits people look for are honey production and gentleness. And then another thing that's sort of becoming more and more popular is having bees that are resistant to diseases and pests. There's lots of different types of resistance available. You can buy, Purdue makes these mite biters, which you might have heard them called mite maulers, ankle biters. They have all these different names, but the trait is that they damage varroa mites, and you can find them on the bottom board with missing legs like the one in this picture. There's other bees that can have hygienic behavior where they're pulling out pupae and mites when they're reproducing, which is also correlated to having some resistance to chalk brood. That's what you can see in the bottom right. There's other bees that are VSH, or varroa sensitive hygiene, where they can recognize that the mites are reproducing, open the cell, stop the reproduction of the mite, and close it. And that's another trait. So there's various things that people look for when they're looking for a stock. So we solicited the help of beekeepers because we wanted to have a large sample size of these queens being assessed. And we also wanted it to be a blind study. So these people were provided with the queens for this study. The queens were marked particular colors, and they had no idea, the beekeepers had no idea which line was which. They just knew that there were queens with an orange dot, pink, yellow, blue, or green. Each beekeeper in Pennsylvania had 20 colonies dedicated to this study for 2 years, and they each had 20 colonies, so they had 4 of each of the 5 lines. In Indiana, they had trouble finding enough cooperating beekeepers, so 3 of their beekeepers had 50 each, a 20 and a 30. So it wasn't ideal. It was, I think the way we did it in Pennsylvania was easier on the beekeepers because we really asked them to do quite a bit of extra work that they would not have normally done. So I'll show you the timeline in a minute, but basically the beekeepers were asked to assess the colonies every single month from April through October. And then I would go and visit them for 2 of their assessments. So I would go for a spring assessment and work alongside them, and a fall assessment. And I think it was a really nice sort of outreach piece because, you know, working side by side with a beekeeper that you respect is really nice. And I learned a lot from them, and I think some of them learned from me. And yeah, it was just, I really appreciate them. And they, like I said, did a lot of work that they would not have otherwise done. So here you can see a representative of each of the lines. And I'm now allowed to reveal which line is which. Up until this spring, the beekeepers were in the dark about that. But we do have a paper that has been submitted for publication, and so it's going to come out there. So we had blue and green were our sort of controls. So these are queens that are brought to Pennsylvania by the tens of thousands every year. When I think of packages, I think about Georgia, but I learned that Man Lake brings in tens of thousands of packages from California. So both of these lines or stocks are being brought to Pennsylvania every spring en masse. So many of you probably have Georgia or California genetics in your colonies. The other 3 that we chose to look at were 2 local stocks. So pink was our Pennsylvania local. These were sort of tried and true Pennsylvania hardy bees. Indiana had the same. Most of the beekeepers during the study assumed that we had, you know, real Purdue ankle biters in the study, but we did not. These Indiana, they did not claim to be ankle biters, but they were, you know, good local Indiana hardy stock. And then there was also a Russian line from, a certified Russian breeder in Indiana. So one of the interesting things that came out of this is we're sort of interested in beekeepers' perception when looking at a queen. So I'm guessing that when you're just looking at these pictures, you might be like, ooh, look at that juicy yellow one in the middle. That's my favorite. Or, ooh, look at that black one from California. I love those black queens. But really, does the color of her exoskeleton mean anything? Um, anyway, it's just I'm just throwing that question out there. I want you to think about your perception of what you're seeing already. So the beekeepers clearly could see the queens, right? So they probably had preconceived notions. They could talk amongst themselves and like try to guess. But really we wanted them to just analyze what they're seeing, and we did accept their feedback in a survey at the end. But it was super fun talking to them and them trying to get out of me which one was which. So this project started in the summer of 2020. It's a miracle, but all the queens had to be shipped. I made sure even the Pennsylvania local had to be shipped to Pennsylvania because shipping is a big deal. And they all had to arrive on the same day. And it actually happened. It was like a major miracle. And so I received all of the 200 queens for Pennsylvania, and I numbered them, and I split them off into this beekeeper, that beekeeper, that beekeeper, that beekeeper, and then I drove them where I needed to get them to the beekeepers. Then it was up to them to requeen their full-size colonies with these queens. Those colonies had been treated for mites with Formic Pro at least 2 weeks or more before the queens arrived, because we wanted to make sure that they started off with low mite levels. So they were requeened in June. And then when I came to visit in September, we assumed that the turnover of the workers was such that the workers present were from this particular queen. So they would act the way this queen's genetics were. And when I took a sample, it would be her, daughters. So when I visited, we did colony assessments. We broke them apart looking at temperament. I took a sample for lab analysis. We did a varroa wash, and we always tried to find the queen and take a picture of her. One thing that I think happens a lot is a beekeeper claims that, like the paint mark came off, but it's still the same queen. And I have serious doubts that that's still the same queen. So I always wanted a picture, show me, so I can put them side by side, which I'm about to do, and see, is that really still the same queen that the beekeeper thinks it is? Because some of these colonies at the end of April 2022 were claiming still the same queen. So anyway, just out of interest, we had to find her, take her picture. Then the real meat of the study was during the 2021 season. That's where the real field work and assessment by the beekeepers occurred. And then we did a second winter, so the 21 to 22 winter, and then a final sample was just taken in April, and then they were free to do what they wanted with those colonies. So one of the things that we measured was temperament, and basically that's sort of like how gentle are these bees. So we came up with this 5-point scale, and I now use it all the time on my data sheets. So the scale is 1 to 5. 1 is the nicest, calmest bees you've ever seen. You wouldn't even need a veil or smoke. They pretend you're not there. All the way up to aggressive, you're getting stung all over, you need a full suit, smoke is basically not helping. Those are the bees. You may have heard that I was looking for mean bees this summer. The 5s are what I was looking for this summer. But I don't want to keep 5s, and I didn't want the queens we were assessing to be 5s either. But each time the beekeeper went in, they were supposed to mark from 1 to 5, how mean did they seem? And I understand that it's seasonal, it has to do with skunks and dearth and all these other things. That's why we measured it a whole bunch of times. We also, of course, measured honey production. So not the first year when we requeened the colonies, but that second year, that full year when that queen was in place the whole time. Um, uh, Charlie Voracek was one of the collaborators, and this was his very favorite colony. You can see how tall it is already in May when I visited in 2021. They have since, grafted from this colony. They just love this colony. And uh, it was interesting to me to see the organizational skills of the different beekeepers. And Charlie has this daughter, Shanna, who's like this ultra, you know, organized person. So she has every box labeled with the color and the number. And that was my very favorite to work with, because I knew that they were keeping track of everything very closely. So you might be getting a hint here about some honey production. Of course, we also did alcohol washes, and that was every month. And the beekeepers were asked to treat an individual colony if it got over 2%. So they weren't doing this treatment on a apiary basis as some people do, because this is research. It was every colony individually washed and treated as needed. And then, of course, we took some samples for lab tests. So those had to be either collected on dry ice, which is like minus 80 or so, more than minus 80 degrees, or on alcohol and in a cooler, and then brought back to the lab, and the abdomens were dissected and crushed and looked at for viruses and gene expression, and we'll talk about that with the results. So here's our first results. This is the average honey extracted from the boxes of each line. And I'm sure everyone is like blinded by the yellow bar because it's so bright and so tall. Those Indiana bees were fabulous honey producers, followed by the Pennsylvania bees. So they were dramatically making more honey than the others. In fact, also the yellow were very good survivors over that first winter. I don't have a survival slide. I think I forgot to collect that information. But anyway, you can see the local bees collected more honey that year. These data are the brood area from when I visited. So I visited the first fall, then the following spring, and then that fall. So when I visited, when we went through the colonies looking for the queen to take her picture, we also estimated the brood area. So number of frames of brood. So a 10 frame deep box could have a total possible of 10 frames of brood. And what I see here is those yellow Indiana bees were kind of going gangbusters early in the spring. So this was like the first spring assessment for the year. And surprisingly, the Russian bees as well. I always think of them as, having a smaller winter cluster. So how could they have so much brood? But they did. I haven't run statistics on this. I don't know if it's statistically significant. But just looking at these raw data, most of the time it didn't really matter which line, but this yellow bar stands out to me. In terms of temperament, well, it was highly variable. I think it depends on what the weather was like that day. So I don't really hold much to this data. But at least we didn't see like one of these lines being 4s and 5s all the time. So I think they're all equally gentle. And of course, going later on in the year, they get a little more crabby. May they're usually gentler, and I do see that here. In terms of varroa mites, remember we did treat before they requeened, and I asked everyone to try to keep them under control, under 2% whenever they saw it. But in the second fall, you can see that the mite levels were a bit high, and I think some of the beekeepers waited for me to visit before treating. So that's kind of why they were a bit high. They kind of like had to wait for my presence to do their wash and decide to treat. So having it be a bit higher, that could explain it. But I was surprised to see these two package producers having less mites that fall. I have to look back and see, perhaps they got over threshold early and got treated earlier. That would be in my notes. I just thought of looking that up. So it doesn't look like any of these, though, were really, really good at keeping the mite numbers down. They were capable of having high mite loads. So that's a little bit disappointing. What about the lab tests? So you have to remember, I'm a field person. I want to be out with the bees, measuring honey production and varroa mites. So I'll do my best to explain this, but this is why, we have research teams, because some people have strengths and weaknesses. So in terms of those bees that we collected that first fall that were from that queen introduction, her brood, the first, what I'm calling a trial, is Liz Cameron. She was a postdoc. She looked at an interesting research question. You might not care about this, but is there variation within colonies? So a lot of the time we do these conglomerate, mush up a whole bunch of bees and see what is happening at the colony level. But Liz looked at things in individual bees just to see if all the bees have the same level of virus or if it's like highly variable within a colony. So to do this, she looked at 10 individuals from each colony, and she only looked at 3 colonies each from the 5 stocks. These colonies all were from the same apiary, so it was like one of our cooperating beekeepers' samples, 3 colonies from each of the 5 stocks. So this is what she found in that study, and what you should be looking at is the height of these bars. So a taller bar here, sorry, it's not a bar, it's a box. This is a box plot. The median level of DWVA in the Georgia stock was here, but this box is quite tall, which means there was a lot of variation. Okay, so within the Georgia 10 individuals, there was a lot of variation. So some had a lot of virus, some had a little. And that carried over as well to DWVB, the other strain, the worst strain. If you look here at Indiana and Pennsylvania and even the Russian bees, it was less variable. So that's what we're looking at here, intra-colony variation. Liz also looked at 3 other viruses. So we have black queen cell virus, Israeli acute paralysis virus, and Lake Sinai virus. And basically, I'm going to probably repeat this later, but black queen cell virus is everywhere all the time. And IAPV is barely anywhere ever. So that's pretty interesting. And it didn't vary much. Lake Sinai virus, though, was highly variable in the Indiana stock. So all of this information together is just a very short paper that we submitted that basically says viral titers are highly variable among the individuals in a colony, and some stocks have more variation than others. So when a researcher is deciding to go ahead and do a project where they're measuring some of these, they need to consider which stock are they working with, and also, is it okay to mash 10 bees together and get one number? So this is just about to be published, and so we'll see what the response is by the research community. But we decided to go ahead with mashing the bees together for the next trial, which is there variation between stocks? So of course, that's what we're trying to get at here with this study. That trial 1 was kind of like a bonus study within our study. So I want you to really remember, and since this is being recorded, the data will probably change by tomorrow, or at least next week. So this is not final anything. This is still preliminary. But for these tests, the lab work takes 30 bee abdomens and crushes them all together. And so far, Charles, who I mentioned, Charles Dean, has gotten through 9 colonies each from Georgia and California, and 6 colonies each from Pennsylvania, Indiana, and the Russian bees. And I think we're going to do 30 colonies of each. So this is just sort of the beginning, the first few bits of information. So for this trial, thinking about the lab tests to run, because we saw black queen cell virus kind of everywhere, we consider this to just be endemic to Pennsylvania. It was in 95% of the individuals that were tested. And so we cut that out of the second trial. And also because IAPV was so very uncommon, we decided not to measure that either. But in their place, we added some interesting genes, and I haven't talked about genes yet at all, but people have found genes associated with hygienic behavior. So unfortunately, we did not do hygienic testing, but we were able to look for the genes. And also, there's another gene, neurexin, that is telling about mite biting behavior. And while we did do mite biting assays, I don't have the data yet. And the data I looked at from Indiana, that not enough mites fell onto the sticky board to do the assessment. So if you know about mite biting assays, you put a sticky board in under your screen bottom board, and you let the mites naturally fall or get groomed off by the bees, hopefully, because they're, mite biters. And then you collect that and you take it back to your lab and you remove each mite very carefully, look at it under the microscope, and determine whether it's injured or it's just intact and fell. And you can't have a proportion of mites bitten unless you have at least 10 individuals. 100 is much better to find. But the numbers I saw for Indiana were rarely 10. They were 3s and 6s and 8s. So we can't use the data, unfortunately. So what did we find so far with these mashups of 30 bees? So what you can see here, and I'm sorry that the colors are in a different order. So before orange was over here and yellow was here, so they're flip-flop now. What you can see here is preliminary results on which colonies had more deformed wing virus. And for DWVA, these results look a lot different than what Liz showed. But by the way, none of these graphs that you can see right now or the next graphs have any scientific significance using statistics. Usually with statistics, we use a value of 0.05 as our maximum number to say they're different. And some of these are at 0.058, but we can't say that they're significant yet. So the story remains to be seen as we have a larger sample size. So it does look here like there is numerically some variation in how much DWV these colonies have. In particular, what I see is the DWVB levels in the California bees does seem to be higher than the others. Notice that the Pennsylvania one here was quite low and also not very variable. So looking at neurexin, this is your mite biting. Totally strange results. I would have expected, even though the Indiana bees were not official mite biters, I would have thought they would have more of this neurexin, the mite biting gene, and it does not look like they're any different than the others. And then in terms of being hygienic, again, kind of all over the place. However, I want you to note, you might look at the graph on the right and be like, ooh, look, the Georgia and California bees have more hygienic. But no, these lab people, if this gene is overexpressed or higher on the graph, it actually means they're less hygienic. So you kind of have to think in reverse here. So this higher level is actually less hygiene, and this lower level is more hygiene. So pulling out the mites and the bees when they have them. So I'm sorry, I feel like I'm sounding very confusing, but the picture is still being painted. So just to summarize, please remember this is preliminary. These graphs will change. They will never be published because we're going to get, many, many more bits of information. So far, no significant differences, but they're close. So if things continue the way they are, we should see some differences, or it could go the other way and become less significant. We will see. So I don't feel like we're giving you a lot of great information to tell you which stocks to purchase or like avoid packaged queens or anything like that yet. I will tell you, if you want to know which beekeepers were involved in the study, Jeff Eckel is here tonight. He was one of the participants. They did have their favorites. I heard a lot liking the pink and the yellow just before they even knew where they were from. A couple liked the green and blue. So, it just goes to show that, as always, ask 10 beekeepers, you get 11, 12, 20 different answers. So what are we doing next? We have begun a new trial, very similar, only this time I'm the one keeping the bees and I'm the one blind to the breeds. So they're all Pennsylvania breeds, except for I also purchased some Georgia queens just to have that as a control. I requeened my colonies this summer, and so I'm managing them blind. I only know the colors to compare them. So we'll see what comes of that. But really, what we really want to be able to provide you guys is data-driven recommendations for which bees will perform the best here. There's a lot of momentum around, local is better. And, we need the data to show that. So we're going to keep looking and studying and assessing these bees. So, rest in peace, Queen Elizabeth. We will always remember you when it comes time to remember which color to paint our queens. And I just want to thank all the participating beekeepers that did all the hard work behind keeping these colonies for us. And also, this was funded by the USDA. So we thank them for that. And that is my talk. Questions, please.
That was great. So anybody who wants to ask a question, use your little reaction button down there and raise your hand, and that will keep it a little bit organized. Norris, you look like you're raising your hand in real life.
Muted. Still muted. Well, while you figure that out, I'm going to look at the chat. Someone asked if the queens were mated in their source area. Yes, they were. Oh, Norris, go ahead.
That's my question.
Okay. Yeah, we solicited the breeders. We asked them to make all these queens all at the right time for us. We allowed it to be later in the year because, we wanted them to be able to do their commercial business first and then, service us with care. We also asked them not to bank them for a long time and to put them in a particular cage, and they came in all kinds of different cages. So that was a little, you can give the protocols to the beekeeper, but you can't. I got to come up with a good pun about that, how they don't listen. Anyway, so yeah, they were mated. They were not instrumentally inseminated, so they were mated in place. Yeah.
Great.
And my question was did the Indiana bees actually produce all that much more honey in Pennsylvania?
Yeah, I should have told you all of those pretty rainbow graphs were all Pennsylvania only data.
Oh, okay.
Yeah.
Wow. I want Indiana bees. I screw the local stuff.
Sure. Yeah. They made a lot of honey, man.
Yeah. Any other questions? This is a great study. Jeff, are you
He looks like he's
indicating you
trying to
want to
unmute.
ask a question? There we go.
Hey, Jeff.
Well, my question, I guess, is maybe a little off topic, but when was the first alcohol wash data? And you probably don't have this information right at your fingertips, but I'm curious how well the formic worked across the board. Was that was the only treatment used or the first treatment used 2 weeks before?
That's what they were every beekeeper was asked to do. We did not, I don't think the first official varroa wash was until August or September.
Okay. And the cleanup, if they use formic to clean up, that would have been in July probably, or I did it myself.
I was like, why are you saying they? No, June.
Oh yeah, right.
Yeah. No, you
June,
must have, I
okay.
think you were asked to do a varroa wash every month.
Yeah,
right?
I think so. So I'd be curious how well the formic worked. If you could pull that data at some point, that would be interesting to me.
Yeah. Shouldn't you have checked that before the queens
Yeah,
came?
well, but I'm talking about 25 apiaries across multiple states. I don't have that data.
Is it statistically significant? I think that's what we're looking for, Robyn.
Right. I don't know.
Justin.
Don't know yet.
I apologize. I was driving. How much more honey did the Indiana bees make comparatively? Did you have a certain percentage to that, or just weight on average?
I can get that number. Right now I would be just guessing based on the graph. But so what I see here is like 130 pounds for Indiana. Pennsylvania was more around 80. So right there, and that was the next highest, and the others were at 50 or less. So like almost triple, well, certainly triple what a Georgia or California queen made.
Now, that speaker who stepped in and filled in the slot last year at the state conference, she seemed to reference that out in the Indiana area at the higher elevation seemed to be the best area for honey production. Do you think there's any relation there that the bees or bees that come from a more prairie environment, a more meadow environment, possibly are better foragers? Do you think there's any kind of relation there with the geographic location and the bees' behavior?
I don't know, but we just got a grant to study foraging behavior. So maybe we will figure that out. We better consider our genetics when we start next spring.
It looked like, I mean, Justin, as you said, you were driving. There was another chart that showed differing levels of brood, and to me, it looked like the Indiana bees made more brood than the others, and so it may be less foraging behavior than it is timing in the way that the volume of the queen's laying.
Interesting thought. So when we were going through the protocols, like we would meet with the beekeepers and discuss, okay, what are we going to do in the spring with these colonies? Like, because a normal beekeeper would like probably do a split, right? And we're like, no, you're not allowed to do a split. We need this queen and this hive, And so they came out of winter at different strengths. And absolutely that could have affected their honey production. So I need to talk to the cooperating beekeepers for this Pennsylvania study. I think we're going to take a different approach, and we're going to start them all off in spring at the same strength. So we're going to, mess with them that way, like make them all nukes starting fresh or something like that, so that it's more genetics and less strength. Right, Jeff? I think you were, advocating for that.
I had one that really came through weak, and a frame of brood from a strong one would have really took that from doing nothing all year to, potentially making a honey crop.
Yeah, research is constraining.
I get it.
How did you control for swarming, or what is there swarming data in here?
They had to check them at least once a month. So, and then if they saw, some of them went back time after time after time, cut out swarm cells, cut out swarm cells, cut out swarm cells. And others were not as, attentive. So I do have, whether the original queen is present each time because of those pictures and everything, and data about swarm cells. But that's also probably a confounding factor. Yep. So I think going forward with the Pennsylvania study, if we do, take the queen as a nuke, start her fresh, we shouldn't have to deal with any of that, hopefully, if they have enough space, right?
I have a question for you, Robyn.
Yep.
At Penn State, how do you, as well as the people at UC Davis and in Atlanta, recruit people to do your research in this stinging environment? Because it's amazing to me that there's anybody who, aside from bee nuts, who really want to do this. Are they part of some graduate program that they're involved in, or what is it that gets them to come to you?
How did I get Jeff Eckel to help me?
Well, he's nuts. We know that.
Well, we just have a relationship with people around the state. I mean, it's a really big, my job requires a lot of networking, right? So that's why I'm really excited to have this reception with club presidents. And I want you guys to network with each other. It's all about, talking. So we didn't want to ask anybody who only had 20 colonies to put all their colonies toward our research, right? So there's a limited list, of sideliners available. And so, we think about their personalities, whether they're actually going to possibly listen to me and follow my protocols. That was a big one. And other collaborators were like Mark Gingrich. I call him first every time, Charlie Voracek all the time. I did call Steve Rapaski, but he refused to do double deeps because he's the single brood chamber management guy. So he found me another guy in his area. I also wanted it to be geographically spread, all across the state, so that I had to be like, hey, Brad Seward, you're awesome. Do you have a friend that's awesome? And you're in the Northeast, And so it's all kind of word of mouth and making phone calls.
And how do you get your graduate students to involve in this?
I don't.
There must be somebody there who's writing the papers and doing all the crap work.
No.
Is just
No.
you and Jeff? I can't believe that.
Just me and Jeff. I mentioned the people that did the lab work. Those were postdocs that we hired. I don't have a lot of graduate work. If you heard about the comb study, we did have one graduate student get her degree off of that study, studying the microbiome of the bee bread. I don't know if you heard about that study. That paper's about to be submitted too. So that kind of thing happens, but it's more, I've been just really kind of doing it myself with the beekeepers. So, and I hire beekeepers that live near my colonies as assistants. I pay them hourly to come out with me on the days when I need 3 people or whatever. Did that answer your question?
Yeah, sort of. I'm surprised that there are no, entomology PhD students who are involved in this.
But they're too busy taking classes. Like, this kind of research is just too time consuming, too much travel. We have a brand new project I mentioned that's going to be looking at organic farms and organic management, and then looking at where the bees fly, just to see if, on a landscape scale, if they're staying within the areas without pesticides and stuff like that. That's going to have a few graduate students. But our lab, we're hiring a postdoc. So that's somebody with a PhD it's like a step between your PhD and getting a faculty position. And it's a time for them to get like more papers and, some leadership skills. So that's what we are currently advertising for at Penn State. There's going to be a master's student at Virginia Tech who is going to study like waggle dances, and at Cornell there's going to be a master's student. So it's coming, but I mean, I don't know who's on here, but students are difficult. They have classes that go till May, and you're like, dude, you got to get out in the field. The bees are doing their thing. So it's just a little frustrating.
Wow.
I'm being too honest. Wait, shut the recording. It's honesty. Yeah.
Well, I'm impressed.
Oh, thank you. I like having my hands at the bees. I never want to be stuck in my office like telling other people how to manage the bees. I want to be there too.
Well, thank you for a great presentation. I think we've learned a lot and been inspired. Maybe we'll come and do research for you.
Sure. If anyone wants to volunteer to help me manage my bees, you can come out anytime. Or if if you're someone with 100 colonies and you can dedicate 20 to a study, could probably use your help.
I had a question. Are you going to tell us who the queen breeders were that you got the queens from?
Can you send that to me in an email? I'm not positive if I'm allowed to do so.
Okay.
Yeah, I think even in the paper, let me look at the paper that's about to come out and see if it's in there. If it isn't, I'll just tell you. So do you want to know? Okay, sorry. I'm gonna think before I speak.
From like the Indiana and the Pennsylvania one.
Jeff, do you know? I'm just curious because if you know, then I can probably tell. I think you do. You must. Jeff's still there?
He's
I don't
there, but
ca-
he's not talking.
I don't believe I know.
So probably none of us can know.
Or I just don't pay attention.
That could be. I'm looking at the paper, what we wrote about. We did tell who Georgia and California were. Yeah, I just send me an email, please. So I hope I'll see a bunch of you at the state meeting. Come in time for the icebreaker because it should be fun.
Indeed it shall. Indeed it shall. So any other questions, or I mean any sort of general beekeeping questions or other things anybody wants to bring up?
I have another question about queens. Last year and this year, I raised a queen like late in the season, and they never laid one egg. So last year I left her in there a long time, and the hive pretty much dwindled out to nothing. This year I combined it, I just got rid of the queen and combined it with another hive because I didn't want the same thing to happen again. Have you seen anything like that, or have any ideas why that could be?
If you're specifically asking me, she didn't get mated properly is my first guess, especially if it was late in the season.
Yeah, that's what I was thinking, because it was right during the really hot spell that she would have gone this year anyway, that she should have gotten mated. But
How late was late in the season, Priscilla?
trying to remember, it was the last round of queen cells that Scott was doing. So I'd have to look at my notes, but probably sometime in August.
Yeah.
Yeah, I would recommend, looking up some sort of queen rearing calendar because, if she's not laying by such and such a day, you should just go ahead and replace her. You know what I
Yeah.
mean? Like, you don't want to get into a laying worker situation, which is so
Right, it
difficult,
was,
right?
she definitely should have been laying, like I gave her plenty of time and I added more brood and did a bunch of different things to try and stimulate her to get her to lay, and she just never did.
Yeah, I guess
Did you,
she taught you.
did you feed it all? I have bees in like bluebell that once the nectar flow ends, like they don't get much, if anything. And if they shut down completely if I don't feed. So
Yeah, I
would
tried that
imagine,
too.
okay.
I didn't feed like a lot, not as
Yeah,
much as I should have, but I did.
some stimulative feeding might be a good solution, or at least worth a shot. I would think there's probably like freak things that can happen too if oviduct got clogged or something like that. Do you know how to tell if they're mated? Like if you pinch the queen, you can pull the spermatheca out, and if it's clear, then she doesn't have any sperm in there. If it's cloudy, then at least she mated. I have a
Well,
drone
I
layer.
saved her in alcohol, so I
Okay.
can
Are you
try
going to the
that.
state meeting?
No,
Were
Okay.
you gonna say bring her? We'll look under the
Yeah.
microscope.
Well,
yeah,
I'm going to bring mine. I have a drone layer that I had to pinch. So I threw her in alcohol, and then I thought I'd pull the spermatheca out at the state meeting for somebody. I was talking to Frank about it.
Priscilla, if you want to get it to someone and have someone look and take photos for you, we could probably do that.
well, maybe I can give it to Jeff. I'll drop it off at his house and he can bring it.
Perfect.
Sounds good. Make sure you label it with your name so we remember.
Okay. But I was just wondering if anybody else, like, if that's a common thing going on. I guess not.
No.
I have had, queens that didn't lay, and yeah, but it's I didn't notice any particular problem this summer as opposed to any other time. I don't know if I mean, we did have that long, hot, kind of dry spell for a while. I don't know if we had a talk last month about drone congregation areas. I don't know if hot weather reduces mating, is bad for mating or not, but we did have, it was a really kind of hot and dry summer. We have a question from Mr. David Mora. Go ahead.
Yes. I had some problems at some point this season with little hive beetle. And of course, they got into my honey supers and of course left this gooey liquid behind. Is there something I can do? Can I use those frames again, or is there something I have to do to those frames? Do I have to put new foundation or what in them?
Yeah, the bees won't clean up that honey, but did you have plastic foundation? If you just scraped it, you could reuse it, certainly.
Oh, I certainly, yeah, some of it was plastics foundation, yes. So scrape off the wax, the comb down to
Well,
the
because the honey
foundation.
is fermenting, that's what's happening there.
Yeah,
So
yeah,
you want to just like get rid of the honey
right,
for
right.
sure.
Okay.
I wonder if you can rinse, if you might be able to rinse it out or something, depending, maybe not. Like it certainly would be easiest just to scrape it and re-wax it if it's plastic foundation. But if the comb's in okay shape, you might get away with.
Well, what if
keeping the comb.
what if I cut off, well, I'm sure some of it's uncapped, but if I cut off the cap and centrifuged it and then washed it, like you said, you think it would be usable then, or just that's
Like-
too much work to do?
Yeah, I mean, and it's nasty, so like you don't necessarily want to put it in your extractor, although it might be fine.
Okay, good.
Uh,
Safety
but-
be-
Yeah, it depends. Like they can kind of chew all the way through the foundation, not plastic foundation, but they can like destroy the comb, but they don't always, or at least most of the comb is in good shape. And I kind of always tell beekeepers that your drawn comb is the most valuable thing that you have.
Right. Oh, definitely.
But
Yeah.
Robyn's kind of right that it's like
Start
the
over.
bees don't want to eat it. It's
Yeah.
not good, so you gotta clean it up best you can.
All right. Thank you.
My tip for that would be I invariably take too long to extract my harvested honey supers. And when I get to the bottom of the stack, I'll have some wax moths and small hive beetles. So if it's bad with the small hive beetles, I'll put it in the freezer, kills all the larvae, right? And then I'll put it out back, and the wasps and the bees, even this time of year, will clean it up pretty good. And then next year, they seem to reuse it and repair it just fine. That's my experience.
That's actually really interesting because I do find the yellow jackets do like fermenting stuff much more than honeybees. So if you let them at it, they'll still be there long after the bees are not coming anymore.
Mm. And my experience has been like Justin's. Like there is sometimes when a beetles really slime a hive out, there's certainly a lot of comb like in the brood nest that is kind of just crawling with beetle larvae that you have to trash. But if the stuff is, if it's still if the comb is still recognizable as comb and it has honey in it, and it's just like looks greasy from the fermentation, that stuff I will freeze, and they will, bees and whatever will clean it out. So I don't know. I think I wanted to hear what everybody else was saying, because I've just done what I've done kind of by instinct. I don't know nobody ever taught us how to deal with this stuff. But I think there is stuff that they will clean out. But if it's obviously just, there is stuff that's just too gross to even consider.
I think like best management practices with small hive beetles, something we should be discussing, because it seems like a lot of people leave dead outs just out, on the hive stand, and often beetles will reproduce in that. So I feel like we're often doing things to kind of shoot ourselves in the foot, or just be a nuisance to the rest of the community.
Lost a lot of honey.
Yeah, no, it's devastating. Yeah, it can be. They're a serious pest, and if you're like financially dependent on that would be a really big loss.
One thing I always do, and I got an upright freezer free from a neighbor who was just giving it away, and so when I harvest, I will
Here.
put 4 or 5 supers into that freezer. So I harvest into the freezer, freeze everything for 2 or 3 days, and then then I can stack it, and as long as I don't let stuff get in there, it stays good for quite a while. But I don't. But that's the way I deal with it, and that's specifically that's my best practice for beetles, because I hate having any bugs in my honey.
And when you freeze, I've heard it's not a problem, and I trust that it's not a problem. But my gut reaction is like adding water content to the honey or something, like when it thaws out or anything. Are you bagging it? Are you letting it sit, warm up, obviously, but do you run a dehumidifier? Do you do anything special? Any reason to think that there's something to what I'm saying?
No, I don't. I haven't had any problem with it. A little bit of moisture will recondense when you pull them out if they're cold, but I haven't had any issue with it. And mostly I will either keep them in plastic bins with lids on them, or I'll stack up supers and keep them covered. Either one. And I don't have any trouble with extra moisture or anything like that. You get a little bit of honey kind of leaking out of some of the frames, and that kind of drains out. And if too much drains out, it's a mess. But that's the worst of it.
Yeah, I thank goodness have Steve Fink and his Bethel freezer at my disposal for a slight fee, and I always freeze stuff beforehand. We've used brood minders to see how long does it take to thaw. It doesn't take long. Last year all kinds of things happened that we delayed, and it was like January, February, and I'm like, can we please finish our extracting from last year because I'm paying for this stupid freezer. But it was fine. It's always fine. Can be in there 2 days or 6 months or more, and it's still liquid, still extracts.
I have not had any trouble with the freezing and thawing it. Unlike everything else in beekeeping.
It's the one problem-free area.
Uh,
The problem is people don't have freezer space. That is the biggest problem. Maybe we
That's
need
right,
to
you have
invest
to-
in some, giant freezer facility rental every year for people. I don't know. Club freezer.
It's not a bad idea, but I don't know that we'll be doing it. I mean, there are, of course, refrigerated trucks that will freeze stuff, and that's a pretty effective way. You could rent one of those for a few days. Anyway, any other questions? Any other observations? Nobody will mind if you talk about something other than queens?
Dr. Jamie Ellis gets that question every once in a while on his Two Bees in a podcast about, is it okay to extract honey from frames that might have wax moths or small hive beetles? And he said, oh yeah, the commercial guys, there's definitely a certain percentage of that in everything that's getting extracted. So there's invariably that's going to happen, and it's harmless. But I had asked him, well, you know how when maggots eat a wound, they excrete things that's beneficial and medicinal. Might the slime from small hive beetles have maybe some kind of beneficial medicinal property we can market a little bit? And he didn't have any information on that, but he thought it was a good question, and it could have possibly some validity. But I thought it'd be a funny marketing technique, like with Don Chump and his Doom Bloom honey from the spotted lanternfly. Maybe we could do something with the small hive beetle slime outs.
Are you going to start sampling it, Jason or Justin? Are you going to how are you going to pursue this? Because
well, that's where I draw the line. I draw the line at the smell test. When a frame's getting slimed out, and if you put your face up to it and it reeks, yeah, get rid of it, put it outside, definitely freeze it. But if it doesn't smell bad, and there's a lot of small hive beetles, throw it in the freezer, kill the beetles or pick them out. It's a little bit cathartic. Get a nice little flat, straight tweezers and pull them all. I feed them to my chickens. I don't know why. Maybe I'm OCD or something, but I pull them all out, put in a little container. I'll take them up and feed them to the chickens the next morning. So they don't go to waste. Even the frozen ones that fall out in the freezer, I'll collect them and feed them to the chickens as well.
Well, Justin, I'm sure you guys have heard, because Jeff is the one who told me that someone sent their Doom Bloom off for analysis, and it's like more medicinal than Manuka. So I have sent off some samples for further testing of the lanternfly honeydew honey to see
Great.
if that stands.
Cool.
Well, no, I did not hear that. What I did hear on the Hive Jive podcast out of Texas, they brought in a Turkish doctor. I don't know if it's Farouk. I think it's Dr. Farouk. And he was doing some studies down there comparing the light and darker honeys. And he was finding in the fall honeys, the darker honeys, more of that measurable medicinal component that's in Manuka. And then a year or two later, John Swan, the host of the podcast, mentioned they talked to Dr. Farouk again, and he found that buckwheat honey in the States has more of the medicinal measurable component than Manuka does. I did hear that about buckwheat. I didn't hear that about the Doom Bloom spotted lanternfly honey. So that's interesting as well.
Yeah, I'm gonna put the website in the chat.
it's actually Japanese knotweed, but who's counting?
What do you mean?
Oh, I don't know. We had 1 year where we had a lot of where the honey tasted odd and different. And I haven't had that kind of honey. We have a lot of Japanese knotweed honey in the fall around here, But around here, I haven't tasted the lanternfly honey since
Aren't
then.
you lucky? So lucky. Do you want some? Because I made quite a bit this year. And hopefully your knotweed flavor and color are just like overriding what the lanternflies are doing.
Yeah, maybe. I don't know if anybody else agrees or disagrees with me. But I don't think it tastes like that one year when we had a lot of lanternflies.
I dropped off fall samples when the spotted lanternfly was near its peak during the one state conference, and I don't remember getting any results back from that. So I have no idea what that.
Yeah, that's me. That's my fault.
What did you
I'm
find
still
from
working
that?
on it. What's that?
Are you still on it? Okay.
Still on it.
Oh, I dropped off honey to that thing too. So yeah, we'll see. That the honey I dropped off was the year that it tasted like lanternfly to me.
Well, I still have some. You want it back?
I definitely do because I sold out on that stuff.
Well, I could bring you a pail to the state meeting. It'll only be $250.
I'll take it.
Now, Robyn, does yours taste odd? I never really had any of the fall honeys in my area taste funny at all.
Oh my gosh, you guys still need to know what this stuff smells like. I will bring samples to the state meeting. If you need one, let me know. It smells bad. It smells smoky and has a horrible aftertaste. That is my opinion. Some people like it. I do
I
not.
think I have the sample that you did the taste test with at the comb project thing or wherever
Really?
it was, or maybe it was the
Yeah.
state meeting. So if we ever meet in person, I'll bring that. We can all taste it.
Yeah.
Honey tasting sometime.
I have some fresh
if you
2022 jarred stuff in my house.
Yeah,
if you
okay.
can bring it, I would
You
love
can
to
come.
try it, Robyn. I'll
All
trade
right,
you.
and I also have my knotweed from this year, so I'll bring that too, so you can see and taste the difference. It's like one's red and one's like brown.
And you have some hives that get knotweed and some that get lanternfly?
Yep, I have a knotweed site, and my lanternfly site is unpredictable because so this is what happened. I have bees in Burnville, which is near Steve's place in Bethel, and usually fall they get nothing, right? They're the ones I have to end up feeding. And so this year I was like, let's put all the wet supers from all the sites at Burnville. They'll clean them up, and then I can take them to my knotweed site later. They filled them with honeydew. I was so mad because not only do I have honeydew honey, but I didn't have supers to bring to the other place where we get beautiful knotweed. So it was like a double whammy.
That lanternfly stuff is really high in whatever it is.
We don't know yet because I haven't told anyone. It's high in poop. Anyway.
Well, we try to work a little bit of lanternfly into every meeting. I'm glad we accomplished that this month. Any other questions, observations, unreasonable requests? All right. Well, I guess that will wind things up. Thank you so much, Robyn Marie Underwood, for coming and sharing your wisdom with us. And thank you to everybody else who joined us. It's been swell. Have a great evening and eat
Thanks
more
for having
honey.
me.
So long, everyone.